We will now continue our discussion for general biology tutorials. We are now in module 2, which we will talk about touring the cell or the cell components. What will we talk about in this video? So we will talk about microscopy, we will talk about cell theory, the primary types of cells, and the different cell organelles that make up cells of living organisms. So first, let us talk about microscopy. So why is it important that we talk about microscopy? Because it is through microscopy that we are able to determine and we are able to visualize what is inside our cells. We are able to observe what are the functions of different organelles, and we are able to differentiate the functions of different cells from different parts of the body of an organism. So we have different types of microscopy that help us understand the mechanisms behind the function of different cells and its organelles. So we have first light microscopy, where invisible light is passed through the specimen and then through the glass lenses. Next, we have the electron microscopy, wherein a beam of electron is focused through the specimen or onto its surface. Next, we have the scanning electron microscopy, wherein you will coat your specimen with thin sheets of gold or gold foil, wherein this gold-coated specimen is exposed to an high-energy electron beam. And lastly, we have transmission electron microscopy, wherein we will use an electron beam that will cross through a very thin section of the heavy metal stained specimen. So kung sa scanning electron microscopy, we will coat your specimen with gold. In transmission electron microscopy, we will coat our specimen or we will stain our specimen with heavy metals such as lead and other heavy metals. So, ano bang pinagkaiba-iba nila? So, they differ in their ways of mechanism, gaya nga ng mga binanggit ko kanina. And they also differ kung ano yung tinitingnan natin. Whenever we want to visualize the specimen as a whole, we can either use light microscopy or electron microscopy. However, if we want to visualize the surface of a cell or the surface of an organelle, ang titingnan naman natin dito is the scanning electron microscopy. On the other hand, kapag naman ang titingnan natin is yung surface or yung inside, yung interior ng isang cell or interior ng isang organelle, kumbaga titingnan natin yung cross-section ng isang organelle or yung specimen natin, to be specific, we will use transmission electron microscopy. Now, let us move on to the development of cell theory. So, alam naman natin ngayon that the classical cell theory has 3 tenets. And pag-uusapan natin yun mamaya. And let us first explore kung ano ba yung mga events na nangyari bago mabuo yung mga tenets of cell theory natin. And titingnan din natin kung sino ba ang mga relevant persons na pwede nating alalahanin or pwede nating tandaan kapag napakinggan natin ang salitang cell theory. So first, we have Robert Hooke. So ano ba ang contribution or ano bang ambag ni Robert Hooke sa cell theory natin ngayon? So isa siya sa mga pinakaunang tao na nag-aral tungkol sa cell because he termed the coin cell after observing box-like structures when he observed cork. So yung cork, alam naman natin, yun yung nakikita natin na takip sa mga bote ng wine. And these cork are made up of dead plant tissues na pinatigas. So when he observed cork, he observed box-like structures. So para siyang ganyan. So ganyan na magkakadikit. So he observed these box-like structures with the use of crude lenses. So wala pa siyang microscope noon. Hindi pa siya yung microscope na alam natin ngayon. Ang microscope lang nila noong araw is crude lenses lang. And through those crude lenses nga, nakita niya yung cells ng cork. So, saan ba nagmula yung term niya ng cell na yan? So, he coined the term cell from the Latin word cellae, which mean cubicles. So, makes sense naman, di ba? Kasi ang nakita niya nga is box-like structures. And he named them cells because the Latin cellae means cubicles. So, yon. So, ito yung isa sa mga pinakaunang events that led to the development of cell theory. punta naman tayo ngayon kay Anton van Leeuwenhoek. So, he is known as the father of microbiology. Bakit? Kasi isa siya sa mga pinakaunang nag-publish ng studies kung ano yung mga natuklasan niya tungkol sa isang cell. Further, isa siya sa mga bumuo or isa siya sa bumuo ng pinakaunang microscope na kahawig or resemblant dun sa microscope natin today. Kasi si Robert Hooke, ang ginamit lang niya is crude lenses. However, si Anton van Leeuwenhoek, ang ginamit niya na talaga is a microscope na sarili niyang gawa. So, kumbaga, it is a fully operational system of lenses na, hindi lang basta crude lens. Okay, so that's why he is known as the father of microbiology. So, using those single lens microscopes na ginawa niya, he was first to observe gherkins or animalcules, which turned out to be bacteria and spermatozoa upon further studies. So, yung gherkins is a foreign word. I'm not sure if it is kung anong klaseng lengguwahe siya, to be specific. But this word is translated into English na naging animalcules. So, animalcules means tiny animals. So, yun. So, yung animalcules, yun yung una niyang nakita through his microscope. Kasi ang samples na pinagkuhanan niya ay nanggaling sa isang taong may sakit ng gout. So, nakita niya doon, kung nakita niyang parang samples, na tinawag niyang animalcules dahil wala pa namang studies relating sa cell na nagagawa noong panahon niya. So, he called them gherkins or animalcules in English. And upon further studies niya, napag-alaman na yung palang mga nakita ni Leeuwenhoek is bacteria and spermatozoa. And then we have Henri Dutrochet. So, ano naman ang relevance ni Henri Dutrochet? Kasi typically, ang mga naririnig lang natin pangalan sa cell theory is si Hooke, si Leeuwenhoek, si Schleiden, si Schwann, and si Virchow. So, what is the contribution of Henri Dutrochet in the development of cell theory? So, through observation of cells in animal tissues, so eto na. So, kung kanina, si Leeuwenhoek, ang tinignan niya is fluids or samples from a deceased human. Dito naman tinitingnan niya is cells in animal tissues. So, he concluded that a cell is a fundamental element of organization. Dahil nga sa limited technology no ang mga panahon na yon, nakita lang nila na there are structures within the animal tissues. However, they are not able to differentiate or definitively identify kung ano yung mga structures na yon. So, kumbaga, it was still unknown at the time. However, Henri Dutrochet concluded that yung mga little structures na bumubuo sa animal tissues, yun daw ang fundamental element of organization. Kasi hindi mabubuo ang isang animal tissue kung wala yung mga maliliit na structures na yon, which turned out to be cells. So, he also claimed that cells are not just structural units, but also physiological units. So, what does he mean by this? So, ang ibig sabihin niya naman dito, hindi lang daw nandun yung cells para mag-provide ng structure sa katawan ng isang organism. Andun din daw yung mga cells to carry out physiological processes. Or, sa madaling sabi, yung cells, ito yung parang hint na cells are the basic unit of life dahil ang pagiging physiological unit ng isang cell means that a cell has the ability to carry out basic life processes, which kung halimbawa nito yung mga homeostasis, regulation, then yung mga discharge of waste products, intake of nutrients, and among others. So next, punta na tayo dito sa lagi silang magkasama pag pinag-uusapan natin yung cell theory. So lagi silang partner, si Matthias Schleiden and Theodor Schwann. So ano ba ang mga contributions naman nila? So nauna si Schleiden. In 1838, Schleiden defined the cell as the basic unit of plant life. A year later, Schwann followed suit, defining the cell as a basic unit of animal life. So, yung studies kasi ni Matthias Schleiden and Theodor Schwann ay independent of each other. So, hindi sila magkakilala and they are from different parts or different countries and yet they manage to have the same conclusions. So, ang pinagkaiba lang, Schleiden focused on plants and Schwann focused on animals. However, kahit magkaiba sila ng pinag-aralan, they were still able to conclude that the cell is a basic unit of life for either plants or animals. And therefore, this proceeded to the second tenet of our classical cell theory. So dahil sa mga discoveries nila na to, kaya nabuo yung second tenet ng cell theory, which is cell is the basic unit of life. Next, we have Robert Remak. So, Robert Remak is suggested as the original proponent of the idea that cells come from pre-existing cells. So, kung matatandaan natin, this statement that all cells come from pre-existing cells is heavily attributed to Rudolf Virchow. However, upon historical studies na pag-alaman na there is a possibility that Virchow plagiarized Robert Remak's idea dahil si Robert Remak ay nauna nang mag-publish ng kanyang statement na all cells come from pre-existing cells. Then si Virchow naman ang nakilala nga sa statement na omnicellula is cellula, which is just a Latin phrase meaning for all cells come from pre-existing cells. So, kanino natin ngayon i-attribute ang third tenet ng cell theory, which is the all cells come from pre-existing cells. So sa ngayon, it is still a debate kung sino nga ba dapat ang i-credit natin kung sa statement na to, si Robert Remak ba or si Rudolf Virchow. However, either way, kung sino man ang napagdesisyonan na kung kanino i-credit yung statement na yon, yung development na yon is still a major step towards the formulation of the cell theory. So, ngayon nakita na natin kung sino ang mga primary and relevant scientists and people who developed the classical cell theory. So, we can summarize their developments and their findings into 3 statements. So, ito yung classical cell theory natin. Number 1, all living organisms are made up of one or more cells. Which explains the existence of unicellular and multicellular organisms. Next, we have the cell is the basic unit of life according to the findings ni Henri Dutrochet. And lastly, and according din pala to sa findings ni Matthias Schleiden and Theodor Schwann, that the cell is the basic unit of animal and plant life, respectively. And lastly, we have all cells arise from pre-existing cells, which came from either Remak or Virchow. So sa ngayon, di ba, as technology progresses, meron na rin tayong mga innovations and recent developments na na-observe pa natin sa cells. And kumbaga, kailangan na rin ng update ng classical cell theory. That is why we have added modern tenets of the cell theory. So, ano ba mga nakapaloob naman sa modern tenets of the cell theory? So, first, energy flow occurs within cells. So, alam naman natin that energy flow is present in nature. It is a unifying theme of biology and is a characteristic of life through energy processing. So, tama. Kasi alam naman natin, gaya nga nasabi, cell is the basic unit of life, kaya niya mag-carry out ng basic life processes, and since energy processing is a characteristic of life, therefore, ang cells kaya din nila ng energy flow. So, yon. And then we have heredity information is passed on from cell to cell. So, another characteristic of life which stands for reproduction. So, as living organisms reproduce, nagkakaroon nga ng genetic diversity, and yung heredity information from the parent is passed from the offspring. Another process that can explain this tenet is the occurrence of mitosis and meiosis. And last modern tenet is that all cells have the same basic chemical composition. So yon. So these 3 additional tenets were found through our improvements in cell technology and different breakthroughs in cell research. So here's an activity for you to try out. So these are 8 statements, true or false lang. Assess whether the statement is true and or assess whether the statement is false. So this is a kaya nyo na yan, independent activity na yan, kung gusto nyo sagutan. If okay lang din namang hindi. So we have the first and the second statements here. So nilagay ko na rin yung answer key sa may parting ilalim if you want to check out for yourself. So yan. Now let us move on to the different types of cells. So we have 2 types of cells, which are the prokaryotes and the eukaryotes. Prokaryotic cells can be classified or can be branched out into 2 main organismal domains, which is the archaea and the bacteria. And eukaryotes naman, isa lang ang domain niya, and lahat ng eukaryotes ay nakapaloob sa domain ng eukarya. So, punta muna tayo ngayon sa prokaryotes. So, prokaryotes, this mainly consists of bacteria and other unicellular organisms. So, bakit siya kailangan pa ng dalawang domains? Bakit hindi na lang natin pagsamahin? So, ang pagkakaiba kasi nila, bacteria, ito yung mga typical bacteria na nakikita natin. For example, we have E. coli. Famous example, E. coli, Streptomyces, Staphylococci, and other types of bacteria that are typically found in nature. So, doon nakapaloob si bacteria. Doon nakapaloob yung mga organisms na yun sa bacteria nakapaloob. Yung mga organisms naman na nakapaloob sa domain archaea, are extremophiles. So, ano ba yung mga extremophiles naman? So, extremophiles are organisms that can live on extreme environments. For example, sobrang alat ng tubig, kayang mabuhay pa rin ng isang halophile doon. Kapag naman sobrang init, meron pa rin mga organisms na nabubuhay doon, which you can call thermophiles. So, yun yung mga examples ng extremophiles that are classified under the domain archaea. And kapag naman domain eukarya ang pag-uusapan natin, ang mga nakapaloob naman dito is lahat ng mga multicellular organisms na nakikita natin, such as plants, animals, fungi, protists, and among others. So yun, so lahat ng mga multicellular organisms, doon sila nakapaloob sa eukaryotes. So first, let us talk about the similarities between the prokaryotes and eukaryotes. So every cell daw, either a prokaryote or eukaryote, all share 4 commonalities. So una, lahat daw sila merong outer covering called plasma membrane. Then they also have cytoplasm in which other cellular components are found. They also all have DNA, kahit prokaryote or eukaryote yan, meron silang DNA, which is the genetic material of the cell. And lastly, they also have ribosomes, which are the protein synthesis sites of the cell. So first, let us define what is a prokaryote. So prokaryotes, their cells do not have membrane-enclosed internal compartments. So ito yung primary feature na hindi natin makikita sa prokaryotes. Wala silang compartments. Kumbaga, kapag tinignan natin ng isang cross-section ng mga organelles ng isang prokaryote, nakasabog lang sila doon. Further, they also lack a true nucleus. Makikita natin mamaya dahil ang nucleus lang nila or region kung nasaan ang genetic materials ay tinatawag lang natin na nucleoid. So, ibig sabihin wala silang true nucleus and are oftentimes they are also unicellular. Gaya nga ng nababanggit ko kanina. So, the first cells ever to form were undoubtedly similar in organization to modern prokaryotes since they appear much simpler in internal structure. So, this just means that yung mga sinaunang life, the first life that appeared on the earth, are highly similar to the organization of modern prokaryotes. So, ibig sabihin, kung ano yung nakita natin sa prokaryotes ngayon, most likely, ito din yung mga nakita natin sa ancient life or yung mga first life na nakita, primordial life to be specific, na nakita natin dito sa Earth. So, ano ba mga examples ng prokaryotes? Gaya ng sabi ko kanina, such examples are Escherichia coli and Staphylococcus aureus, which are two of the most common bacteria that we can encounter in our daily life. So, here is a diagram of a prokaryotic structure. So, meron siya iba't ibang parts. We have the pilus, the capsule, the cell wall, the plasma membrane, the nucleoid or simply the DNA chromosomes. Then we have the cytoplasm, the flagella, and ribosomes. So, pag-uusapan natin yan isa-isa. So, here are the different parts of a prokaryote. So, first, let us talk about the flagella and pili, which are all about the locomotion. So, these are the locomotive parts of a prokaryote. So, ano ba ang flagella? So, flagella is a whip-like structure. So, para siyang lubid or para siyang tali made of protein called flagellin, which provide motility to the cell. It spins on its axis like a propeller, driving the cell along. So, ibig sabihin daw yung mechanism ng flagella para mapagalaw yung cell is umiikot siya na parang ganito. So, yun. So, since it is a whip-like structure, pwede siya maging lubid na paikot-ikutin ganyan na umiikot sa kanyang axis. So, yon. So, ito yung nagpo-provide kung bakit nakakagalaw from one place to another yung isang prokaryote. Next, we have the pili or pilus for the singular term. So, ano naman ito? So, these are thread-like structures that help bacteria adhere to one another during mating and during attachment to animal cells. So, kung si flagella ay para sa movement, si pili naman ay para sa pagkakapit. Pagkakapit niya, kung halimbawa meron siyang gustong kapitan na isang organism, it is through pili or pilus kaya siya nakakakapit. So, these thread-like structures adhere to the surface of the cells that it wants to cling on to. So, yun. So, etong dalawa na to ang isa sa mga pinaka-importanting parts ng prokaryote dahil kapag wala ito, it is very impossible for them to actually live. Now, punta naman tayo ngayon sa protective parts ng prokaryote. So, kung kanina pinag-usapan natin is yung locomotion, punta naman tayo ngayon dito sa protective parts. So, first, we have the capsule. Please take note that not all prokaryotes have capsule. So, pili lang or there are some species of prokaryotes that has a capsule. So, what is a capsule? It is an exterior coating of many prokaryotes consisting of a slime layer made up of glycocalyx. So, yun. So, ito rin tatandaan natin sa capsule. It is made up of glycocalyx and not all prokaryotes have it. Next, we have the cell wall. So, itong cell wall, ito naman yung hindi rin lahat ng bacteria ay nakikita natin. Hindi lahat ng bacteria ay meron pa rin ito. So, what is the function of a cell wall? It is highly similar to the cell wall of a plant cell in terms that it surrounds the cell membrane and gives the cell shape while preventing it from bursting. Kasi syempre, meron din itong internal pressure na minemaintain sa loob, and kapag walang cell wall, due to the differences in pressure of the environment and the internal pressure of the prokaryote, pwede siyang pumutok. So, what is the cell wall of a prokaryote typically made up of? So, organisms under the bacteria domain has a peptidoglycan cell wall. So, ito naman yung ito yung nagiging basis natin kung bakit tayo meron tinatawag na gram-positive bacteria and we have the gram-negative bacteria. So, it is true that this peptidoglycan cell wall which determines the behavior of a cell, whether it is a gram-positive bacteria or a gram-negative bacteria. Ngayon, napag-usapan na natin kung ano ang mga nasa locomotion parts and protective parts ng isang prokaryote. Pag-usapan naman natin ngayon kung ano ang mga nakikita sa kanyang interior. So first, let us talk about the nucleoid. So ito na yung sinasabi nating walang true nucleus ang isang prokaryote. Instead of a true nucleus, it has a region called nucleoid. So it is a region within the cytoplasm where the cell's circular DNA molecule congregates and floats. So, kumbaga, hindi siya compartmentalized and hindi nakahiwalay or hindi nakaseparate yung nucleus niya or yung DNA niya within the interior of the cell. Kumbaga, palutang-lutang lang din yung circular DNA molecule niya within the cytoplasm. Unlike sa eukaryotes na yung nucleus niya is heavily separated from the other organelles of the cell. And next, we have the inclusion bodies. So inclusion bodies are porous protein aggregates that contain organic compounds such as starch, glycogen, or lipid and acts as food reserves. So yon. So typically kasi for example, yung lalo na yung mga molecules or rather organisms that live in extreme environments which are called extremophiles. Typically kasi ang food source nila doon is very scarce. Therefore, they are forced to have food reserves in order to maintain their nutrition and of course to carry out their metabolic processes. So yun, so napag-usapan na natin kung ano ang mga parts na unique sa isang prokaryote. Ngayon naman pag-usapan na natin ang isang eukaryote. So ano ba mga nakapaloob sa domain ng eukarya? So if you recall, gaya ng sabi ko kanina, animals, plants, fungi, and protists have cells that are usually larger and structurally more complex than those of the prokaryotes. So, ito yung mga nakapaloob sa domain eukarya natin. The kingdom Animalia, kingdom Plantae, kingdom Fungi, and kingdom Protista. So, ayun. So, yun yung mga kingdoms or mga types of organisms na nakapaloob sa domain eukarya. So, eukaryotic cells generally have dimensions 10 times greater than those of prokaryotes. For example, a spherical yeast cell has a diameter of 8 microns or 8 micrometers. So, yun. So, ibig sabihin lang ito, talagang mas malaki ang eukaryotic cells compared to prokaryotic cells. Why? Because they are structurally more complex. So the compartments in the cytoplasm whose interiors are separated from the cytosol by a membrane distinguish eukaryotic cell functions. So ito yung nawawala doon sa prokaryotes natin. Sa eukaryotes, meron kasi siyang compartmentalized system na, for example, sa compartment na to, isa lang yung specific function na ginagawa nila kahit iba-iba silang organelles. So sa compartment A, for example, ito yung command center ng cell, yung compartment B, energy processing, then yung compartment C, protein synthesis, and compartment D, yung naman paglilinis ng cell, and among others. So, yun yung wala sa prokaryotes. So, makikita natin yun sa mga organelles na pag-uusapan natin. So, here is a diagram of a eukaryotic cell and specifically an animal cell and a plant cell. So, yon. So let us now talk about the parts of a eukaryote. So first, we have the nucleus. So ito yung nawawala din sa prokaryote natin kanina. So now, instead of a nucleoid, we now have the true nucleus. So, pauna pala, the first 4 organelles that we will talk about is we can call it the central compartment. So bakit siya tinawag na central compartment? Dahil ito ang isa sa mga pinaka-importanting organelles of the cell and it also maintains kung ano ba yung nangyayari within the interior of the cell. So, kumbaga, ito yung parang command center nga. So, unang-una dito is yung nucleus niya. First, it contains a majority of the cell's DNA and the work of the nucleus is to dictate to the cell what to do, whether to grow, divide, or die. So, ayun. So, ito yung parang brain ng isang eukaryotic cell. So, ito yung nagsasabi kung dapat na ba siyang mag-grow, dapat na ba siyang mag-undergo ng mitosis or meiosis, or dapat na ba siyang mag-undergo ng cell death or tinatawag din nating apoptosis. So, yan. So, additional knowledge lang. Ang tawag po natin sa programmed cell death or parang cell suicide is called apoptosis. Then, we have the nucleolus. So, nucleolus is a structure within the nucleus where components of ribosomes are assembled. So, nucleolus naman, kumbaga parang mas malaking part naman siya or specialized part siya sa isang nucleus. So, ribosome assembly naman ang kanyang function. Next, we have the nuclear pores. So, nuclear pores are made up of large complexes of proteins and allow certain molecules to pass through the nuclear membrane or the nuclear envelope. So, ito naman yung parang mga security guards ng nuclear membrane, kung ano ba dapat yung pumasok and hindi dapat pumasok sa nucleus ng isang cell. And it works hand in hand with the nuclear envelope, which is a double membrane layer that separates the contents of the nucleus from the rest of the cell. So kung nuclear pore ang mga security guards natin, yung nuclear envelope naman, ito mismo yung pader na binabantayan ng nuclear pores. So yon. So napag-usapan na natin ngayon kung ano ang mga central compartment ng isang cell. Pumunta naman tayo ngayon sa protein synthesis compartment ng isang eukaryotic cell. So first, we have the rough endoplasmic reticulum. So, the endoplasmic reticulum kasi sa isang eukaryotic cell has two parts, which is the rough and the smooth. And ang pinagkaiba lang nila is that the rough ER or the rough endoplasmic reticulum contains ribosomes, while the smooth ER does not have any ribosomes. So ano ba ang function ng isang rough ER? So as a compartment, it segregates certain newly synthesized proteins away from the cytoplasm and transport them to other locations into the cell. So ito yung nagka-classify kung ano yung mga functions na gagawin ng isang protein na binubuo na ni ribosomes. So habang ang isang protein ay nasa rough ER din, pwede pa silang modify chemically so as to their function, and eventual destination can also be altered. So, punta naman tayo ngayon sa smooth endoplasmic reticulum. So, the smooth ER does not have ribosomes and it is more tubular. So, ang structure kasi ni rough ER is typically para siyang flattened sacs. So, si smooth ER naman is more tubular and less like flattened sacs naman. So, within its lumen or within the interior of the smooth ER, proteins can also be chemically modified. So, ibig sabihin lang yung mga sinisynthesize ni ribosomes na proteins ay nag-undergo ng continuous chemical modification para ma-suit or ma-appropriate kung ano yung magiging function niya and kung saan siya pupunta. Either within also the interior of the cell or kung ipapalabas ba siya ng cell as an extracellular product. So another function of the smooth ER is that it is also the site of hydrolysis of glycogen in animal cells as well as the synthesis of lipids and steroids. So yung hydrolysis of glycogen is very important for energy processing, and the synthesis of lipids or lipids and steroids is very crucial for the maintenance of the phospholipid bilayer of the cell membrane. Next, we have the Golgi apparatus, or also known as the Golgi bodies. So, it is made up of cisternae, which is the collection of flattened membranous sacs and small membrane enclosed vesicles. So, yun yung bumubuo sa cisternae. So, what is the role of the Golgi body? So, its role is to receive the proteins from the ER. So, ngayon nakikita na natin kung ano ba nagiging pathway ng isang cellular protein inside the eukaryotic cell. So, una, pupunta muna siya sa ribosome kung saan siya nabubuo. Then, pupunta siya sa rough ER or rare for simplification. Then, pupunta naman siya ngayon sa SIR or the smooth ER. And lastly, pupunta ngayon siya sa Golgi apparatus kung saan ngayon siya pinapackage, kinoconcentrate, and sinosort before they are sent to either cellular or extracellular destinations. So another function of the Golgi body is that the polysaccharides of cell walls, which are observed only in plant cells. So yan, so plant cells ang meron yan ng cell walls. So yung mga polysaccharides ng cell walls daw, gaya ng cellulose and starch, are also synthesized in the Golgi apparatus. Lastly, next we have the lysosome. So ngayon nakita na natin yung protein synthesis or protein packaging compartment. So ngayon punta naman tayo sa digestion compartment ng isang eukaryotic cell. So first in line, we have the lysosome. So lysosome is a membranous sac of hydrolytic enzymes that many eukaryotic cells use to digest macromolecules. So hydrolysis din yung ginagawa nito, parang yung naunang organelle natin kanina, which uses the hydrolysis of glycogen. So dito naman, hydrolysis or hydrolyzation of macromolecules naman ang ginagawa niya. So yung hydrolytic enzymes ni lysosome and its membrane are made by the rough ER and then transferred to the Golgi apparatus for further processing. So yun yung basic function ni lysosome, and we can see that this digestive compartment is also highly linked with the protein synthesis or protein packaging compartment na nakita natin kanina, which includes their ribosomes, rough ER, smooth ER, and Golgi apparatus. And next, ito naman si mitochondria, alam natin, powerhouse of the cell. It belongs to the energy processing compartment of the eukaryotic cell. So ano pa bang mga organelles ang kasama sa energy processing compartment ng cell? So dalawa lang sila. It is the mitochondria and the cell chloroplast, which are, as we all know, observed only in plant cells. So mitochondria is the powerhouse of the cell, and its primary function is to convert the potential chemical energy of those fuel molecules. So ito na yung nangyayari, yung pinaprocess natin yung glucose into a form that the cell that can use, which is the energy-rich molecule adenosine triphosphate or ATP. So the production of ATP in the mitochondria uses fuel molecules and molecular oxygen is called the cellular respiration. So ayan. So ito lang yung ginagawa ni mitochondria. Wala siyang ibang ginagawa kundi sa cell kundi mag-provide ng energy and to carry out the processes of cellular respiration. Next, we have the peroxisomes. So peroxisomes are organelles that collect the toxic peroxides such as the hydrogen peroxide or agua oxinada, the H2O2, that are the unavoidable byproducts of chemical reactions. So since nagkakaroon niya tayo ng hydrolysis of and hydrolytic digestion of macromolecules, there is bound to be toxic peroxides as byproducts. And isa pa sa mga toxic peroxides na nililinis ni peroxisomes are, example nito is yung SOD, is the superoxide dismutase, which causes enzymatic browning on plants and causes the rapid aging of skin cells naman sa humans. So, this peroxisome is typically only found on animal cell. However, a structurally similar organelle called the glyoxysome is found in plants naman. So, kung merong peroxisome si animal, meron namang glyoxysome si plant. So, these are sites where stored lipids are converted into carbohydrates for transport to the growing cell. So, typically, yung glyoxysome daw ay nakikita lang sa mga growing plant cells. So, yung mga saplings and yung mga patuloy pa lang na lumalaking halaman. Next, we have the vacuoles. So, vacuoles, typically naririnig naman natin siya sa plant cells din, pero pwede rin siya magkaroon sa animal cells. However, sobrang rare lang ng occurrence na ito. So, typically, plant cells lang and protists ang merong vacuoles. And typically, yung vacuoles na to ay malalaki and take up a large amount of surface area and volume within the cell. So vacuoles are membrane enclosed, so filled with aqueous solutions containing many dissolved substances. So nandito rin yun. So yung dissolved substances na to include dissolved gases, dissolved liquids, dissolved macromolecules, and even waste products. So vacuoles serve a number of functions within the plant cell. It includes the storage, the structure, reproduction, and digestion. So, ito yung mga broad functions ng isang vacuole. However, vacuoles can also be specified or specialized according to the needs of a plant. Next, we have the cytoskeleton. So, cytoskeleton naman, ito naman yung exterior part of the cell na, and it is also the protective layer of the eukaryotic cell. So, in addition to the many membrane-enclosed organelles like the mitochondria, the endoplasmic reticulum, and peroxisomes and lysosomes. The eukaryotic cytoplasm also contains a set of long thin fibers called the cytoskeleton. So ito na yung bumabalot ngayon sa kabuuan ng isang eukaryotic cell. So what is the function of a cytoskeleton? So it maintains cell shape and support. It provides cellular locomotive abilities, and some of its fibers track motor proteins. So typically, cytoskeleton is linked with the extracellular matrix, which is used for a variety of purposes such as cell-to-cell recognition and signal transduction pathways. So yun yung mga purposes ni cytoskeleton. The cytoskeleton can be classified into three parts or three components. The three components of cytoskeleton includes the microtubules, the microfilaments, and the intermediate filaments. So if you're interested in learning more about the microtubules, microfilaments, and intermediate filaments, you can read about it on the internet. Or you can search for ebooks, for example, the Campbell Biology, for further references. Next, let us talk about the centrosome. So centrosome is an animal-only cell. So animal cells lang yan. Animal cells. So yan, animal cells lang siya. So this organelle serves as the main microtubule organizing center or MTOC of the animal cell, as well as a regulator of the cell cycle progression. So centrosome is responsible for the creation of centrioles. So which in turn, ito yung bumubuo ng mga spindle fibers which we can observe during the metaphase and anaphase segments or phases of the mitosis or cell division. So centrosome is a major function or is a major contributor towards the success of a mitotic or meiotic cycle. Next, we have the chloroplast. So chloroplast is, as we all know, can be found only in plant cells because they contain the pigment chlorophyll. So this organelle carries out the process of photosynthesis and has chlorophyll and carotenoids that captures light energy. So yun, so meron din siyang iba't ibang parts gaya ng stroma, the thylakoids, and the grana. And next we have the ribosomes, which as we all know, the protein synthesis sites of the cell and are composed of a complex of protein and RNA or ribonucleic acid. So yan, so ribosomes typically nakikita natin yan, pakalat-kalat yan within the cytoplasm, and most of the ribosomes are created in the nucleolus and which are then transported into the rough ER kung saan nakakapit yung mga ribosomes na iyon. And lastly, we have the cytoplasm, which is a thick aqueous, semi-transparent, and elastic semi-fluid present inside the cell where the other organelles float. So, ang tawag naman natin dito sa thick aqueous, semi-fluid mismo is called the cytosol. So, yun yung more specific term for the semi-fluid that can be found in the cytoplasm. Kasi pag sinabi natin cytoplasm, it contains the organelles. When we talk about the cytosol, it is only the elastic, semi-transparent, and thick aqueous semi-fluid. And lastly, another plant cell unique feature is the cell wall. Because the cell wall is made up of polysaccharides such as cellulose and starch and aminopectin, and the cell wall surrounds the plasma membrane of plant cells. It provides tensile strength and protection against mechanical and osmotic stress. It is also through the cell wall that plant cells are able to develop and withstand the turgor pressure kapag ang isang plant cell ay na-expose sa isang hypotonic solution or masyadong maraming tubig sa loob ng isang cell, hindi nagbo-burst ang isang plant cell due to the presence of a firm cell wall. So, ayun, so that is the primary function of the cell wall. Now that we have talked about the various parts of a prokaryotic and eukaryotic cell, tingnan natin what is shared between us. Ano ba ang mga parts na meron kay prokaryote, ano din ang parts na meron kay eukaryote. So, gaya ng pinag-usapan natin kanina, a prokaryote and a eukaryote both have a plasma or cell membrane. It also has cytoplasm. It also has ribosomes for protein production, cytoskeleton for cell structure. And some prokaryotes and eukaryotes also have flagella because yung mga eukaryotes, specifically protists, have flagella. Therefore, meron tayong asterisk, which means ambiguous case because although all prokaryotes have flagella, not all eukaryotes have flagella. So, selected eukaryotic cells lang ang meron nito, specifically those belonging or classified under protists. And lastly, let's also talk about what is unique for us in terms of eukaryotic cells. So plant cells, ito yung mga parts na sila lang ang meron. So meron silang cell wall, meron silang vacuoles, and meron silang chloroplast. And yung animal cell naman, ang unique sa kanilang organelles is lysosome and centrosome. And in some cases, meron din silang cilia for locomotive abilities. And that concludes our discussion for the Module 2 of the General Biology tutorials. If you have any questions, freely contact me on my email or my Facebook account. And that's all, and see you in the next module.